Hello, my name is Genesis Torres Arbildo. Today I'm presenting my research about the impact of the CRISPR-Cas technology on humans. To introduce the topic, in 2012, the CRISPR-Cas9 technology was developed by Professor Jennifer Doudna and researcher Emmanuel Charpentier. This technology has been a great discovery in the science of gene modification because it is a powerful tool for editing genomes, meaning it allows researchers to alter DNA sequences and modify gene function easily. In 2015, the first human trial was done in a human embryo. Though it was successful, the experiment created a high-profile debate about the ethical implication of such work. So there is a controversy about whether genetically modifying human embryos is ethically valid to continue research. The CRISPR-Cas9 technology has been a revolutionary discovery in the genetic engineering field. This technology has shown potential implications including correcting congenital defects, treating and preventing the spread of diseases. Similarly, this technology has given us a tool to change genetic material in living organisms by using a mechanism that was originally a defense system in bacteria. In this video, I will explain how the technology works, as well as what I believe it should be used for. I will also discuss some ethical concerns that this technology raises. A little bit about the background of this research is that according to the website lifescience.com, Nicoletta Nales, she's a doctor with degrees in neuroscience. She suggested in this article that the CRISPR-Cas9 technology system consists of two main components, the CRISPR gene and the Cas9 protein. The CRISPR is short for CRISPR-Cas9. CRISPRs are specialized stretches of DNA, and the protein Cas9, where Cas stands for CRISPR associate, is an enzyme that acts like a pair of molecular scissors, capable of cutting strands of DNA. CRISPR is an acronym for Cluster Regularly Interspaced Short Palindromic Repeats, which are short interspaced sequences of DNA that are repeats of each other. In between these identical strands of DNA, there are different smaller strands of DNA as well. CRISPR technology was adapted from the natural defense mechanisms of bacteria and archaea, a domain of relatively simple single-cell microorganisms. Researchers found that the small sequences in between the repeats were virus DNA. This shows that when virus DNA attacks the cell, the bacteria have DNA on the virus stored or can store DNA. So the complex also holds the DNA for the nuclease protein Cas. This protein can separate a gene and cut it with molecular scissors. So when the Cas protein searches for the DNA, which it has to cut it, it opens up a genetic sequence by separating it and then checks after the DNA matches its RNA. If it reaches Cas, it starts to destroy the DNA by cutting it. If it does not match, Cas closes the DNA again and continues searching for its matching DNA. So when the components of CRISPR are transferred into other more complex organisms, those components can then manipulate genes. And that process is called gene editing. So no one really knew what this process looked like until 2017. But when a team of researchers led by Mikishiro Shibata of Kanazawa University in Japan and Hiroshi Nishimazu of the University of Tokyo showed for the very first time what it looks like when a CRISPR is in action. According to the journal Nature, on an article called CRISPR Cas9 beta globin gene targeting in human hematopoietic stem cells, published in November 7, 2016, says that in April 2015, Chinese scientists used CRISPR-Cas9 technology on human embryos to repair a mutated beta globin gene which causes beta thalassemia. This inherited blood disorder causes the body to produce less hemoglobin, leading to respiratory problems. As the embryos they used were triponuclear, which means the zygotes would not naturally be able to form into a fetus, thus facing difficulties from the beginning. Unclear conclusions may be drawn from these experiments regarding the safety of genome editing in humans. This case is the start of the controversial debate of whether using human embryos as test subjects is ethically valid. According to the journal Nature, on the article What CRISPR Baby Prison Sentences Mean for Research, says that in 2018, Dr. He Jiankui, a biophysicist, announced he had created the first gene-edited babies in the world, which cost him his freedom. Dr. He has a YouTube channel called the He Lab, and in one of his videos, he presented a video called About Lulu and Anna, twin girls born healthy after gene surgery as single-cell embryos. The doctor here explains how he successfully removed a specific gene that prevented the children from getting HIV, which was passed on to them from their father. Dr. He Jiankui safely proceeded with the surgery, taking precautions for the safety and well-being of the mother and the human embryos. He also deep sequenced the twins' genomes before and after birth, just to verify the surgery successfully removed the gene to prevent HIV infection. So now we have to ask the big question. Should the CRISPR-Cas9 technology, a revolutionary discovery, continue to be researched and used in human embryos? So the hypothesis that came out in this research is that if the CRISPR-Cas9 technology continues to be researched in human embryos, then it would have beneficial outcomes that will help future generations. And so harmful diseases such as AIDS and cancer will be cured. This powerful tool will continue to evolve and be able to assist others with new features. From the magazine Scientific American, on the article called The Dark Side of CRISPR, published on February 16, 2021, argues that people like us shouldn't be edited out of existence in some version of a utopian future. This vision of a future without people like us limits our ability to live in the present. So evaluating the quality of life of another person is a complex, highly subjective, and context-dependent task that is morally questionable in a society based on the concept that all people are of equal value regardless of their differences. The limitations of human imagination make it uncertain, if not unethical, for a person to grasp another person's or group of people's quality of life entirely. So, to sum up, an adult can consent to have something modified using the CRISPR method. Nonetheless, there are ethical considerations regarding CRISPR by using it, predominantly without consent. For example, in editing the genome in embryos. These are modifications which change the generations of humans, thus modifications on an organism that affects its evolution. Still, an embryo cannot agree, nor can its offspring that also receive these modifications. Okay, so these are my references. And thank you so much for listening.